This slide here is showing our summary of transcription and translation. I think I showed you this slide before. You can see it's starting in the nucleus. We have the RNA polymerase right here, and the RNA polymerase is making the RNA. Looks like we have a little bit of processing next, where we got the introns being cut out. We got the capping and tailing going on. It goes out through nuclear pore. It finds a ribosome. The ribosome assembles and brings in the transfer RNA and eventually makes a polypeptide chain. So that's transcription and translation. Like I said, make sure you are able to explain those for the final exam and explain the processes, the steps in the processes. So the other thing that you need to know for the final exam is the 6 types of RNA. And I'm going to come back and review these at some point. Like I said, there will be a question based around these. It'll be at least 6 marks, so it could be matching. In the past, I think I did a fill-in-the-blank question where I had the name of the RNA on one side and the function on the other side, and you had to fill in whichever was missing, either the name or the function. So in this unit, in the translation unit, we talked about messenger RNA, ribosomal RNA, and transfer RNA. In the replication unit, we talked about the telomerase RNA. In the transcription unit, we talked about small nuclear RNA. We have not talked about this one yet. That is going to be, we're not going to get to that today, but it's going to be on Friday. We're going to talk about that last type of RNA. So stay tuned. So back to where we left off last day, we were talking about mutations. I wanted to go through this a little bit more carefully and do another practice question looking at a DNA sequence. So we want to look at the implications of what happens when we have mutations. So turns out there's different implications. Sometimes it can be quite crucial and critical to the protein if we have a mutation. And the example I showed you last day was what happens with sickle cell anemia. So if you take a look, we're looking at a gene here, and this gene is for beta globin. So what is beta globin? It is a part of hemoglobin. Hemoglobin actually has an alpha and a beta protein, and this is the beta protein. So we're looking at one codon here, and this codon is GAG when it ends up in the messenger RNA, and GAG is the amino acid glutamate or glutamic acid. So some people have a mutation in this gene, and this is called the sickle cell mutation. So you can see that this codon here in the messenger RNA becomes GUG, which is valine. So this turns into a phenotype where we have a nice round red blood cell gets changed and it has a new shape, a sickle. And so I think I'd mentioned last day, a sickle is kind of like this big blade that the Grim Reaper carries around, and it's used for threshing wheat, basically. So what is going on here? We're changing one amino acid, and we have a crucial change in this phenotype, right? And so these people with the sickle cells, they have sickled cells that are shaped like sickles, and anemia means they don't have enough blood because these red blood cells are very fragile when they break open. And so this is a significant medical condition. So what is happening between the glutamic acid and the valine amino acids? This is a missense mutation, by the way. I'll come back to the definitions in a moment. So I think I showed you this slide and showed you that we have this glutamic acid is changed to a valine, and this glutamic acid is polar and charged, and it's soluble. And the valine, on the other hand, is hydrophobic, and so it's kind of like a little sticky patch. And so these hemoglobin molecules, they stick together, they make these fibers, and these fibers cause these sickle cells. So there's some pictures, other pictures of the sickle cells, and this leads to this phenotype. I also mentioned last day, I showed you this map here and showed you, that this mutation is pretty common in certain parts of the world, or at least where your family originated from. And it turns out, interestingly, that these parts of the world overlap somewhat with the historical distribution of malaria. So malaria is caused by a parasite that infects red blood cells. And so it turns out that people with that have the heterogeneous, so they carry the allele for this gene, it turns out that they're somewhat resistant to malaria. At least they don't have severe symptoms. And so this is why this gene is, at least this mutation, is relatively common still in some populations. So let's talk about these mutations. And I want to talk about the definitions of these mutations. So the sickle cell mutation was called missense. That's a mistake. And I'll explain what these mutations are in a minute. So let's talk about the first type. First type of mutation, the most common type of mutation, is something called a silent mutation. So let's take a look at a silent mutation. There's the wild type. You can see we've got the DNA, we've got the RNA, we've got the protein. So in this mutation, we're going to change one nucleotide. So you can see this one here is changed. And so it's a GC pair, and now it becomes an AT pair. And so in the RNA, this gets changed from a GGC to a GGU. And if I look that up in the genetic code, it turns out it's the exact same amino acid. We have a glycine and a glycine. So this here is a silent mutation. And a silent mutation, it basically means there is a nucleotide change, but there's no effect on the amino acid sequence. So in this case, biologically, it's almost invisible. It's the same protein, all the same amino acids. Silent mutations are the most common, like I said, because these are tolerated and they're not causing anything detrimental to the gene or to the organism. So they're tolerated very, very highly. So a missense mutation, like in the example of the sickle cell anemia, is a little different. So in this case here, we're going to take a look, and you can see what we're doing is we're actually changing this base pair instead, and we're changing it to a TA. So in this case, we're going from a GGC to a AGC. So if you look that up on your genetic code, we are going from a glycine to a serine. So a missense is a mistake. That's what the mis is for. It's for mistake. And we have a one amino acid change. So in some cases, this can be quite significant in the case of sickle cell anemia. In other cases, it's not as significant. It kind of depends on where it all is in the protein. If it's in the active site, that can be really bad for the protein. If it's kind of in another region and you change a polar amino acid to another polar amino acid, it might not be so bad. Kind of just depends on what's going on. But it's a different amino acid, and so it's a missense mutation. So one more definition, a nonsense. So nonsense, no, not sure exactly where they got the word from, but no means no more amino acids. So let's take a look at an example here. So in this case here, we're changing this base pair from a TA to an AT. And so our RNA goes from an AAG to a UAG. And so that means our amino acid, it goes from a lysine to a stop codon. So like I said, nonsense or no for no more amino acids. And so now what we have is a protein that is shortened. And so the technical term for this protein is, we say it's truncated. So by truncated, you can think of like chop down a tree and you make a trunk. So it's a truncated protein. And this can be, it's usually pretty serious to the protein, right? It means that protein is probably not going to be functional anymore because, well, it doesn't have all the amino acids it needs. There are a few genetically inherited diseases that are caused by these nonsense mutations. This one here is a type of muscular dystrophy, Duchenne muscular dystrophy. I think that's how you pronounce it. And this is an X-linked form of muscular dystrophy. That means it's carried on the X chromosome. And so usually what happens is females will often, they could be a carrier, but they have at least one functional copy of the protein. And so it manifests itself a little bit more often in males. And they have a very severe muscular development issue. And you may have seen some of these people, their muscles aren't right. They don't walk quite the same as everyone else. And it's another genetically inherited disease caused by a nonsense mutation. So these three types of mutations here, these are usually what you see in when you see mutations. And like I said, this the most common one here is the silent mutation because it doesn't have any effect. Sometimes though, we do talk about these frame shift mutations. And frame shift mutations basically means you get an insertion or deletion. So let me show you how that works as well. So a frame shift mutation, sometimes you get immediate nonsense. Remember what nonsense means. It means no more amino acids. So let's take a look at an example here. So in this case here, what we've done is right here, we inserted an extra A. So what does that mean? It means that everything gets shifted over. So this TTC, you can see this, the TTC right here, gets shifted over. And all of my codons are now messed up. It's kind of like if you had an essay and I went through your paper and I took out the spaces and added spaces in random places and added a letter somewhere, your words would just, they'd be jumbled. And it's the same thing with the code of this gene is jumbled. And in this case, we've introduced a UAA, which is a premature stop codon. So this is usually really bad and detrimental to the gene entirely. Sometimes you get an extensive missense. So in this case here, you can see what they've done is cut out this base pairing right here. So the A is missing. And so the first couple amino acids here, you can see they're the same, methionine and lysine. Then after that, there's a whole frame misreading going on here. So you're getting all the wrong amino acids. So same thing, this is going to be really detrimental to the protein. So in most cases, these frame shift mutations totally mess up the protein. It's all the wrong amino acids. It's totally non-functional. Once in a while, though, sometimes we get 3 nucleotide changes. And remember, 3 nucleotides is 1 codon. So let me show you an example here. So in this case, we're cutting out 3 base pairs. So we do get it's shifting over, but it's only 1 amino acid missing. So in this case here, if you take a look, methionine is good, and then the lysine gets cut out, but we still end up with the phenylalanine and the glycine. So this can often be not too bad. It kind of depends on where it is in the protein again. If it's in the active site, it could be trouble. It really just depends whether it is a crucial amino acid to that protein. There is one example of a genetically inherited disease that has a frame shift mutation. And this is the most common cause of something called cystic fibrosis. So cystic fibrosis is one of the most common, in fact, I think it is the most common recessive genetic disorder against Caucasians, so people of European ancestry. Apparently about 1 in 700 are carriers. And so I don't know what that means in terms of how many people are in the population with it, but it's common enough. And I've met 1 or 2 people that had a relative with cystic fibrosis. And what it is doing is it is a very crucial amino acid, a phenylalanine is missing. So in this, this protein is actually involved in chloride transport. And so any part of the body that has something to do with secretions is affected. So the gut can have issues, and we can help them with digestion by giving them supplements and enzymes. Where it gets them really bad is the lungs. And so they have a much thicker mucus in their lungs than a normal person. And this can lead to very severe infections. And so unfortunately, people with cystic fibrosis, their average lifespan is 30, 35, because often they will die from a respiratory infection that can't be cleared. So it's, unfortunate there, but maybe someday we'll have some better treatment for it. Okay, so what I want to do is show you what your exam question might look like. So I think last semester I had, can't remember exactly what I did. I had about 10 marks based around looking at a DNA sequence. I think what I'm about to show you is maybe 5 to 8 marks worth of the question. So take a look at this. We've done some of this already. In fact, this might even be the same sequence I showed you before. The first question, like we did last day, is I'm going to ask you to figure out the RNA, figure out the protein sequence. I might ask you questions about which is the sense strand, which is the antisense strand, where is the promoter, where is the terminator. So lots of questions like that I might ask. And then I might ask you about what will happen if we have some mutations. So let's take a look at this. And I'll do this question here. Normally I would do, like I said, I would do it on a whiteboard, but it's a little easier and cleaner for me to do it on the PowerPoint. So the very first thing we need to do is the transcription and translation parts. Okay, and so like I said, you'll have to do this in the final exam, and you'll get marks for this. But so there's the sequence. So remember what I told you before is always start off and look for the start codon. So remember that this here is not the start codon because that is in the opposite direction. Remember, we always read DNA in the 5' to 3' direction. So there's a start codon right there, and that's going to help us to determine all sorts of things, right? So remember the start codon, by the way. The start codon is found on the sense strand, and so this would be the template strand. There we go. So first thing is the messenger RNA. So the messenger RNA, we are going to copy the sense strand, basically nucleotide for nucleotide. Just remember, when we do that, when we copy the sense strand, you are copying and changing all of your T's to U's. So you can see my TAC becomes UAC, and so on all the way to the end. My protein, I'm going to start with the start codon, and you can see what I've done is I've underlined everything in triplets all the way to the end, and I'm just going to read my genetic code. So, and that's going to give you my protein sequence. So AUG is methionine, UAU is tyrosine, CCU is proline, and that is actually the end because, sorry, I'm just getting ahead of myself. There we go. And that is actually the end because UAA is actually my stop codon. So that brings us to the first question I have here in this PowerPoint. It says, what would happen if the T was changed to a C? Okay, so let's take a look at that. You can see I have a little 1 there labeling what it is. And so what I want to do is basically I'm going to make my new codon. I'm not going to worry about the other codons. So my TAT becomes a TAC. So that means my new RNA is a UAC. Okay, so I don't need to change everything else because I'm just dealing with the one codon, right? So if I take a look, I started off with a UAU, and UAU on the genetic code is a tyrosine, and now I'm changing to a UAC. I'm going to circle that on the genetic code here. And so what does that mean? My new amino acid is also tyrosine. So to fully answer the question, I would say that this here is a silent mutation because the even though I have a new codon, I have the same amino acid. So make sure you use this terminology, because when I ask those kind of questions, what would happen? I want to see you use the terminology that we're learning in the class. All right, let's take a look at B. So what does B say? B says, what would happen if the G at position 2 was changed to an A? Okay, so same thing here. I'm looking at my codons, and I'm just going to address this codon here. So this one here is slightly more tricky because I've got 2 strands, right? So it says I'm changing the G to an A, and that means my sense strand is also going to get changed from a CCT to a CTT. So that was a little trickier because I have to change one strand and then I have to change the sense strand. And so just a little bit of an extra step there. From that, I can figure out what my new RNA is. So my RNA is going from a CCU, and now I'm getting a CUU. I can look up CUU on the genetic code. And what is the CUU? It's a leucine. So in this case here, I'm going from a proline to a leucine. So what kind of mutation is that? That is a missense mutation. Remember, missense means mistake. It's the wrong amino acid. All right, so that's A and B. And what is C asking? C is saying, what if we change that T at position 1 to a G? So same exercise as before, right? I'm just looking at this one codon here. And so my new DNA is going from a TAT to a TAG. My RNA, of course, becomes a UAG. And on my genetic code, a UAG is a stop codon. So what kind of mutation is that? This is a nonsense mutation. So if you wanted to be even more complete, you could say that the nonsense mutation leads to a truncated protein, and that would be perfectly accurate and even better than just saying a nonsense mutation. All right, I think there was one more. It was asking D, what if the C at position 3 was changed to a T? Okay, so this is not meant to be a trick question, but it's not as easy as it looks. So let's just go back here and take a look. We've got our start codon here. We've got our next codon, and then we have our stop codon. So that's just the point, isn't it? Is that this particular mutation is actually after the stop codon. So what effect does it have on the protein sequence? Nothing. It's not going to change the amino acid sequence. It may affect something else, maybe it would affect a regulatory component or something like that. But as you can see in my note, that's beyond the scope of this course. But if you want to write that, that's fine. But the answer for this question is it doesn't affect the amino acid sequence because it's outside of the coding region of the gene. So same thing, if I had a mutation, maybe up over here somewhere, it could affect the promoter, but it's not going to affect the sequence of the amino acids. So not meant to be a trick question, just meant to think about what is going on here in terms of the codons and the amino acid sequence. Okay, so a couple other things to mention. There's a lot of stuff in the news about this virus and it's mutating. So what is going on here with these mutations? So it turns out that it's worth it to kind of define one or two things. What is a mutation? Well, all these things we just talked about are mutations. We've got nonsense, silent, and missense mutations. And we're seeing all of them within the virus. It turns out that there's about at least 4,000 variants have been documented in the virus. And we're using the word variant maybe because the word mutant, I think people find that scary and people don't understand mutations. So we don't want to use the word strain because this isn't technically a new strain of the virus. A new strain would mean it has maybe new biological properties or a new host spectrum or something like that. So it turns out, yes, the virus is mutating. That's not a surprise. This is a natural kind of thing. This is what viruses do. In fact, every living thing, as cells replicate, there's always mistakes, always mutations. Some viruses do it really fast. HIV will mutate within one individual really, really rapidly. The flu virus mutates kind of yearly, so there's different flu shot. The coronaviruses, they mutate about maybe 4 or 5 times as slowly as the flu virus. So why are we seeing so many of these variants? Well, it's just a numbers game, right? I mean, you're looking at how many million people and how many millions of infections have happened in the past year. And so you're going to get mutations. And so I want to talk about them for a minute here. So like I said, all those mutations we've talked about have been noticed. Most of the mutations, most of the variants are due to silent mutations. And so you're looking at getting the exact same virus. There's a few that we're concerned about, at least a little bit. Often what we're looking at are the mutations to this gene here. So this gene is the spike protein. So if you remember way, way back when we were talking about topic 20, so that was at the beginning of the semester, I'd mentioned that this spike protein here is important for two things. It's important because the spike protein is involved in the biology of the infection. So it's what binds receptors on human cells and allows the virus to infect that cell. The other reason why the spike protein is important is because for most of our vaccines, this is the main ingredient. This is the main thing that we're basing the vaccine around, because it's on the surface of the virus. Remember the virus? It kind of looks like this. It's got some RNA in the middle, and it's got a protein capsid layer, and then it's got an envelope, and the envelope has these spike proteins. So it kind of looks like this. So this is what the immune system is going to see, is these little spike proteins on the surface. So that's why we're making the vaccine out of that, because you want the immune system to recognize those surface proteins. So the question is, if the spike protein changes, well, what's going to happen to this? So there's been a few mutations that people are looking at. One of the mutations is this one here, this D614G mutation. And so that's a change of aspartic acid to a glycine at a position there. And why do we care about this mutation? We think that this is allowing the virus to be a little bit more infectious. And so this is one of these mutations that we're looking at. And there's some evidence in cell culture that this is the case. Whether this is true in humans or not, we're not really sure. The news seems to be really sure that these variants are more transmissible and more deadly, because this is the news. They want to use the word deadly. The reality is we don't really entirely know. There are some experiments in cell culture that shows that these mutations are maybe making the virus grow a little faster, which might make it more transmissible. There is some data to suggest that this is the case in humans, and that it might be a little bit more deadly. It could be a whole bunch of other things. I mean, think about it. We've been sitting indoors for almost an entire year, eating brownies and cookies, barely exercising. Our immune systems aren't as good as they were 1 year ago, and that's a fact. So there's a lot of confounding kind of variables going on here. So this is why we're calling them variants of concern, meaning that scientists are interested in them and following them, and we want to know what we can do. Is there something we can do different, right? So rather than variant of concern, it sounds scary, but I don't think really anything has changed. I think I have a little table here showing some of the ones we're talking about. These ones here are the three that are getting the most publicity. And you can see this, they have different names. These are the most common names. They all have about 5 different names. The one called the UK variant is one of the ones that people are really shocked at, because it has 23 mutations, 6 are silent. I guess that means 17 are not. And it's believed to be more transmissible. This Brazilian variant is believed to be a little bit more lethal. If you've been following the news, bad stuff is happening in Brazil right now. Yesterday in Brazil, there were more than 4,000 deaths. But they're not locking down in Brazil either. So, is it more severe disease, or is it just, bad policy that's going on, is the question. But, all these things we're tracking because, well, we want to know. We're humans, we're curious, and, we're concerned about these things. So notice the last thing. This is the really big question here, right? Are these things going to make our vaccines useless? And the answer is probably not. There's a lot of good data out that shows that some of these variants are maybe not, the vaccines are not as helpful, right? So I think we talked about before that our vaccines all seem to be about 95% effective, right? So one of the variants, I can't remember which one, there was one small study. So, with vaccines, we need 30,000 subjects to really get good data. But one small study showed that the vaccine was 65% effective. So that's not too bad, because vaccines that are 50% effective are on the market. And it was 100% effective against death. So that's kind of good, right? Maybe the vaccine isn't like amazing like it was before, but it's still 4 stars out of 5. People aren't dying from it. when they have the vaccine and people are not getting severe disease. And so that's kind of the important question. Still, we want to be concerned about these things and really look at the data because maybe we're wrong. These are small scale studies on these variants so far still, and the data is still rolling in. So I just wanted to give you a little information update on that stuff. Again, do what you can, do your part, get your shot, wear your mask, wash your hands, those kind of things. And hopefully we're going to be out and through all of this I'm hoping by September. Apparently there's promises that all Albertans will be able to get the vaccine by July. So we'll see whether that's the case or not. Okay, there's a link for Alberta stats. You can go to, you can just Google that and find that yourself if you want. So we have a few minutes left. I do want to move on and actually introduce the next topic. So let me just pull up that PowerPoint here and we'll get that going. Okay, there we go. Hold on a second. It should be right there with you in a second. Share screen. Okay, there we go. So we have 2 topics left in this class, topic 18 and topic 19, and we'll have some time for review. And what I'm going to do is actually talk about the final exam a little bit on Friday, just to give you a heads up on what to expect. I wasn't going to talk about today because I want to talk about the lab exam. I know you still have that on your mind. So more on the final on Friday. Today I want to talk a little bit about gene expression and regulation. This is kind of one of the final pieces to the puzzle around all this genetic stuff. So far we've been talking about these processes of transcription and translation, and you have these final proteins. So we want to get from DNA basically to biological activity. And so this is what I want to talk about a little bit about how we can kind of tweak this whole system, because of course, we are complex organisms and we've got a lot of things going on. So to kind of introduce what gene expression is, I have this analogy. So this is a challenge, right? Add the punctuation. So people are probably looking at this and, usually this is the kind of thing that happens is the males the men in the classroom say, well, yeah, that's easy. You just put in a couple of commas. A woman without her man is nothing. And the women in the classroom laugh and they say, you got it all wrong. A woman without her man is nothing. So you can see that I have some words, and depending how you punctuate it and how you read it, there is a different context here. And this is kind of like what gene expression is all about, is that we have about 20,000 genes in the human cell, and we want to express them differently in different tissues and different cells. We want to make red blood cells. We want to make cartilage cells. We want to make brain cells. We want to make liver cells. And we want to do this in a way that is going to do things nice and effectively, right? So, this is part of the question, why regulate genes? So some of it has to do with conserving resources. We don't want to build enzymes and proteins that we don't need. Responding to environmental changes. Sometimes, you get a scratch or you burn your finger and you want to heal some tissues. You got to turn on the repair genes. And so environmental changes are a good reason why. Turn on a gene in one tissue, off another. I guess I mentioned that already. Some genes need to be on more than others, right? So we make our skin cells like crazy. And so there's a lot of genes involved with keratin, which is part of our skin cells that are turned on all the time, and other genes are not. So how do we regulate genes? It turns out that this happens at almost every step along the way of all these things we talked about. So take a look at this. You can see this little diagram from the textbook. We've got our DNA, and then we've got our RNA being made, and the RNA gets capped and tailed. It ends up in the cytoplasm. We've got some proteins, and there's there's actually regulation all along these steps. And we're not going to talk about everything. I'm just going to kind of pinpoint and talk about certain things. And most of these are things that we've talked about in some context already. And I want to show you one model for gene regulation in bacteria. Bacteria, of course, are much simpler. They don't have a nucleus and they have a lot less going on. So they're very good models for gene regulation, or at least having a basic understanding of what is going on. So we'll talk about that too. So bacterial cells, as I think I mentioned this before as well, they're super efficient. You can see that as the RNA is being made, we also have ribosomes bind to that RNA and making proteins long before the RNA is actually finished being made. So we have to be super efficient if you're in a bacterial cell, because we don't want to waste a whole bunch of resources. So before, way back, I'm trying to remember when we were talking about, I can't remember which units these were, we were talking about enzymes and we were talking about glycolysis, and we're talking about how you can do regulation by regulating enzymes. So this is a pretty common strategy. So take a look at this. Here's a pathway for the amino acid tryptophan. So you can see we've got 1, 2, 3, 4, 5 genes. And these 5 genes, they make 5 enzymes. Actually 4 enzymes. The 3 enzymes, you can see enzyme 1 is a 2-part. It's got 2 proteins that get put together. So anyway, it doesn't matter. We got 3 enzymes. And we have this chemical precursor right here. And it's going to get converted by these enzymes. So you got these intermediates, and then eventually you get your amino acid tryptophan. So we can regulate this pathway by usually what we do is we turn off the first enzyme. And this is a type of feedback inhibition. So that means that the tryptophan is probably binding enzyme 1 and then turning enzyme 1 off, and you're saving all the materials here along the way. So this is a biochemical regulation, but we can also even take this one step further, and rather than regulate the enzymes, we can save our resources and not even make the enzymes at all. So instead, what we can do is we can regulate these genes and stop them from making the proteins and saving all that energy for making all those stringing all those amino acids together. So what we can do is we can take this tryptophan, and this tryptophan can actually regulate all of those genes, turn them on or turn them off based on what we're trying to do. So what I wanted to do, I was hoping to get to today, was talk about operons. And I think what I'm going to do is kind of introduce the concept of an operon, and then we'll come back and look at this in more details next day. So what is an operon? So an operon, you can see there's my definition. It's the basic unit of gene expression in bacteria. So what does that mean? It means we have multiple genes. So take a look. We've got 3 genes. Now what we're going to do is we're going to regulate them all together. We're going to turn them all on, or we're going to turn them all off. And so how is this done? The first thing to note is that these 3 genes, rather than making 3 pieces of messenger RNA, they actually make 1 piece of messenger RNA. So this is the messenger RNA here, by the way. And that messenger RNA is going to be fed through some ribosomes, and it's going to make these proteins. So then we've got protein 1, protein 2, and protein 3. And what we can do is we can turn this on and off by actually messing with the promoter. So remember what the promoter is. Here's where you need to remember what all these words are, unfortunately. A promoter, so maybe I'll write a little definition for you. So this is a DNA sequence, and this is where the RNA polymerase binds. So it's a DNA sequence, and it's just before the genes, and that's where the RNA polymerase binds. So what we can do, you can see part of the promoter is another sequence called the operator. So I know how many words do we have to learn. Unfortunately, this is the way it is. And if you take some more biology classes that deal with genetics, you are going to come back to operons 100% guaranteed. So it's important to learn a little bit about them now. So the operator is part of the promoter, and it turns out that there is a regulatory molecule that can bind it. So you can block the RNA polymerase from binding and stop this whole thing. So I just want to very, very quickly, oh, there's a slide about the operator. And you can see we have this repressor molecule and it binds the DNA there and it's blocking the RNA polymerase from doing the job. So very, very briefly, I just want to show you this slide. We're going to talk about the tryptophan synthesis operon. So remember the tryptophan synthesis pathway has 5 genes. So here's 5 genes right here. Oh, I circled 4. There's 5 of them. There's 5 genes. And all 5 genes are regulated by this promoter. They make 1 messenger RNA and they make 5 proteins. So I'm going to come back and kind of explain what is going on here next day in a bit more detail. But I feel like it was worth it to at least introduce this to you. So at least you have some of these words in your head. When we come back to them on Friday, hopefully these words will make a little bit more sense. That is where I'm going to finish today. Good luck on your lab exam. I will be available tomorrow during the lab exam over Zoom. If you have any questions or clarifications, you can contact me through Zoom during the time of the lab exam. I will send that out with the instructions when I send out the exam itself, so you can see that in the email. In the meantime, if you have any questions, send me an email or contact me tomorrow morning. I do have a class. I think it's at 10:30 to 12, but I'm available outside of those times as well if you have any questions or clarifications that you have for me. Alrighty then, Wednesday afternoon, that's Biology 107. So I will see you on Friday.